is an... Okay, question 3. The bacteria, Streptomyces, is a microorganism found in soil. It produces a secondary metabolite, the antibiotic streptomycin, which kills other microorganisms. Streptomyces lives in close association with plant roots. These plants produce soluble carbohydrates, which are released into the soil through their roots. Okay, name the growth phase during which streptomycin is produced. So you straight up need to know this. You would get this during the secondary metabolism phase, so that means you're in stationary. Just from a little picture's point of view of this one, we've got our slow, our fast, our slowing, and our steady. Okay, that is our lag, our log, or exponential, our stationary. And if all things continue like this, we would end up into a death phase as well. Okay, so it is this point here where you need to know that you're producing secondary metabolites. Okay, explain the advantage to Streptomyces of producing an antibiotic such as streptomycin. Well, basically, this is going to cut down competition from other things, from other microorganisms, bacteria in particular, from, we're going to say, bacteria, other. Okay, the relationship between Streptomyces and the plant roots is described as mutualistic, suggests the benefit to Streptomyces. Now, you really do need to look at the question properly for this one. Okay, it says that they live in close association with plant roots. These plants produce soluble carbohydrates which are released into the soil through their roots. So that's what you're looking for. The benefit to Streptomyces is that they gain soluble carbohydrates. So basically, they get food, get nutrition from that. Okay, suggest the benefit to the plant. Well, that's got to be about the antibiotic streptomycin. This basically, for the plant, hopefully removes something which could be anything that's a pathogen to it. Gets rid of bacteria, other things. Okay. C. An investigation was set up to compare the effectiveness of streptomycin with other antibiotics by measuring the survival of bacteria. A species of bacteria was grown in the presence of different concentrations of antibiotics, and the percentage which survived was calculated. Results are shown in the table. Okay, we've got a whole pile of antibiotics here, concentration of antibiotics, which I'm hoping even before we get to the question, you're looking at that and going, well, that's a bit dodgy, because they're at different concentrations. And then you've got the survival of the bacteria. Name one antibiotic with which streptomycin could be validly compared. Here's streptomycin. Get my wee highlighter back on this one. Here's streptomycin at 10 micrograms per centimetre cubed. But most of these are not at 10, so the only ones that you could from a valid point of view, compare. Reliability would be how many times you actually did the experiment to get a trustworthy result. Are these two. And you're just supposed to give one of them. Okay, so you can either say it's fusidic acid, or you could give gentamicin. None of the rest are valid to compare because they have different concentrations. Okay, give a conclusion which can be drawn from the results. Now, as soon as you say conclusion, just be really careful that you've looked back up and you've seen what the aim was. Investigation was set up to compare the effectiveness of streptomycin with other antibiotics by measuring survival of bacteria. Okay, so the survival of the bacteria is actually your dependent variable. Our aim is to compare the effectiveness. Give a conclusion which can be drawn. Well, the only ones that we can actually compare to are these ones, because these ones are valid with our 10. Streptomycin, 35% survived, but with the other two, 6 and 5% survived. So conclusion, streptomycin, which you need to actually refer to because it's about that in the original investigation, streptomycin is not as effective as other antibiotics. Okay, conclusions must answer the aim.